Sinai, and as you can see, his talk will be on applying evolutionary medicine to cancer, evolutionary biology to cancer medicine. So, welcome, Bill.
Thank you very much. It's always a real pleasure to come to UCLA to talk about a topic that I don't get the opportunity to talk about very often in my day-to-day activities. I'm a medical oncologist. I'm a breast cancer specialist. But the application of evolutionary biology to cancer medicine has been a very long-standing interest of mine. And as the whole field of cancer genomics has evolved, there's been an intersection now between evolutionary biology and day-to-day cancer medicine, which is really the subject of my talk today. I do want to also disclose that besides being a medical oncologist, I'm actually working in the private industry. I'm the chief medical officer for a breast cancer genomics company by way of disclosure, but I'm not going to be discussing any of their products with you today. Now, I came to medicine already believing in the importance of genetics. I had a master's degree in genetics prior to going to medical school, and that very much affected how I viewed health problems. And in particular, cancer was very clearly a genetic disease as I understood it, and that is, I think, one of many reasons I chose to become an oncologist. And prior to that, of course, my major was evolutionary biology as an undergraduate. So this is the nexus of all of those disciplines. So in terms of applying evolutionary biology to the problem of cancer, I think the most common way in which it has been used, as I'm sure you know, is to explain why cancer develops in the first place. Excuse me. We know that there's an intrinsic mutation rate which is behind the development of carcinogenesis because, of course, mutations are necessary for evolution in the first place. Without them, there would be no evolution, no diversity. So there's an intrinsic risk of carcinogenesis in a multicellular organism that seeks to be able to adapt and evolve. A great deal has been written, of course, about the maladaptation or mismatch issue, where our genome is adapted to perhaps a very different sort of environment and lifestyle than the one in which we're now living. And the result of that is a preponderance of cancer. But what I really want to talk about is a very different topic, thinking about evolutionary biology in terms of how it might help guide cancer therapy, the strategy, in other words, of approach. Although many physicians don't look at it this way, I think it's clear that a cancer in any one individual is really a population undergoing microevolution. And as any biological population will do, it will follow the rules of population genetics. And so what I'd like to talk to you about in the next few minutes is how one might go about applying the principles of evolutionary biology to design and guide a therapeutic strategy. If we were curing every cancer that came through the door, we wouldn't need a new strategy, but clearly we're not. Now we're in the realm of cancer genomics, and this topic is everywhere in the medical literature, and it is also the destination for millions, if not billions, of healthcare dollars and research dollars. We've now gotten to the point where the cost of doing whole genome sequencing or expression analysis on a cancer, a population of cells, or even single cell, has become cheap enough so that it can be done almost in real time to help make treatment decisions. And what's happening now for me as a clinician is I'm actually being delivered the genomic information to try to do something with it to help patients. Now, in terms of where the entire mass of cancer research is going, the National Cancer Institute, as you may know, has launched a multi-year, multi-million-dollar program the Cancer Moonshot program that is really gaining the attention of virtually every cancer center around the country. And where they're putting their dollars is in the so-called NCI MATCH trial, Molecular Analysis for Therapy Choice. That's what MATCH stands for. And it began last year with the intent of enrolling several thousand patients with cancer so that their cancers could be genomically analyzed and a therapy attributed to them, applied to them based upon the genomic mutations in their particular cancer. That's the idea. The idea is that every patient who enrolls has a sample of their tumor sent in to the central laboratory. They look for 143 critical cancer genes that we know are important in driving the behavior of cancer. And once that happens, if that patient's cancer contains a particular mutation against which we have a targeted drug, they will be enrolled in this trial. That's the idea, to prove that you can use genomic technology to decide what to give a patient instead of simply using the same old chemotherapy drugs over and over again for everybody. It's no longer that one-size-fits-all approach. Now, I'm going to go out on a limb and be somewhat heretical and say, I think this is probably not the right approach. And the reason I say this is because they're taking a single gene, single drug approach. That's the idea behind this. And resistance to that one therapy is almost 100% certain when you're dealing with a genomically diverse population of cancer cells. It's an old strategy of therapy that is derived from the way we've been treating cancer, which is primarily with nonspecific therapies like chemotherapy and radiation. But what about the evolutionary strategy? How would things look different if that were being applied? Well, let me just spend 2 minutes on the old strategy, DNA damaging therapy. Doesn't really target anything except the biggest molecule in the cell, the DNA. Kills dividing cells, that's chemotherapy and radiation. It is mutagenic by its nature, highly toxic to even normal cells that are trying to divide. The intent is to induce cell death, and there have been many mathematical models developed in support of how we give chemotherapy, which is what I had been doing virtually every day as a clinician. But it's based on a rather simple-minded concept of biology. The belief is all cells in the cancer are clonally related, they're all proliferating. It assumes they're all identical. And the strategy is let's give as much of these toxic drugs as we can to kill as many cells as we can, but not so much that we're going to kill the host. Let's just take it up to the maximal tolerated dose. And this is the model that's guided therapy now for almost 50 years. And it did work at first. This is a chart showing the success rate in curing childhood leukemia, which was really the first era of chemotherapy. You see the very low cure rate here back in the 60s, 50 plus years ago. And with the use of multi-drug chemotherapy, the cure rate went almost to 100%. And that same strategy was then applied to the more common cancers, the ones that are much more important in human society: lung cancer, breast cancer, colon cancer, prostate, all of those. And it was not nearly as successful. Why the lack of progress in adult cancers with chemotherapy? Well, as a whole, they are much more genomically diverse. The mutational load within an adult cancer is much higher than in a childhood malignancy. So we accept that chemotherapy kills cells by causing random DNA damage and attempting to induce cell death by that mechanism. But for a cancer to actually have become a cancer in the first place through somatic mutations, it already has to have developed a tolerance for mutation. and DNA damage. Otherwise, it wouldn't have survived to become a clinically evident cancer. And therefore, despite the toxicity of these drugs, the DNA-damaging therapies, they don't always kill cancer cells. In fact, resistance virtually always appears in nearly every case of advanced cancer, simply because of the sheer number of cells in that population. In fact, what we're actually doing, which many of my colleagues don't exactly realize, is applying a partial selective pressure and selecting for a truly resistant cell population. Well, thankfully, we're in a new era with a great deal of optimism, so-called molecularly targeted therapy and now immunotherapy. Drugs that are generally less toxic than chemotherapy. Things, for example, drugs that target hormone receptors or growth-promoting pathways, not just DNA, but actual enzymes and kinases that are promoting growth or promoting cellular survival. Really a much more targeted, specific approach that may allow us to target mutations that are only present in the cancer cell population and spare normal cells in the body. No lack of targets. Hundreds of targets have already been discovered. Medicinal chemists in the pharmaceutical and biotech industry have already generated thousands of new drugs. So we don't have any lack of weapons or any lack of targets. What we lack is the appropriate strategy to use them. One byproduct of this genomic era in medicine is that we're redefining cancers based upon their molecular anatomy. That NCI match trial that I mentioned at first doesn't specify whether they're only treating breast cancers or lung cancers. Doesn't matter where the cancer started. All that matters is what is the critical mutation that's driving that cancer, because many of them are held in common from one cancer to another. So redefining molecular anatomy has led to a whole new kind of taxonomy of cancer. And here's an example, the number one cancer, lung cancer. And you can see the various ways in which it has been defined over the years, beginning based upon the cellular appearance under the microscope, to the beginnings of understanding of individual genes. These are genes that are mutated in a large number of lung cancers. But up until a decade ago, we only knew about 2 of them. Now, of course, we've got an expanding pie of different mutations that are telling us that lung cancer is not one type of cancer. It's probably 20 or 30 different subspecies based upon what is driving them genomically. And I want to show you one example of how this has led to a success in therapy that is encouraging many people, but I think falls short of being a home run. So within this group of mutations, there is this one, for example. It's the result of a translocation between 2 chromosomes that produces a fusion protein. It's a growth-promoting kinase that doesn't require any external stimulation to promote the growth of cells. It's a somatic mutation. It's found in only 4% of all lung cancers, referred to as ALK. Once that was found, of course, that mutant protein, which is there in pink in that structural diagram, it was possible to design a small molecule to fit into the enzymatic pocket and inactivate the enzyme. Shut it down, no more growth stimulating signals coming from that enzyme. And then they take this to the clinic. They find a group of lung cancer patients who have this mutation. Remember, it's only 4% of them. You give them this drug. This is a readout of how patients responded. If their tumors shrank, each one of these bars is a patient whose tumor shrank because it's below this line. A very tiny number had further growth. The vast majority responded. This is a CAT scan of how that response looks. And astonishingly, for a disease that normally took people in 6 months, despite giving them chemotherapy, you had people who, you had a 70% response rate in a previously uniformly fatal disease. Fantastic breakthrough. Now, it didn't last very long, however. The result was that you could control the disease for about 7 months on average. That's what this shows here. This is the time course and how long the patients responded before they progressed. And what was found, unfortunately, after that great optimism, was that nearly every patient that responded eventually progressed. They grew through it. Why? Well, it's no surprise if you're an evolutionary biologist why. These are 2 parts of that gene, the ALK gene, both of which affect the binding of the drug. And all it requires is a single base mutation to prevent the drug from binding to the drug. That's it. Now, with a diverse, genomically unstable population of cancer cells, the likelihood that one of them was going to be able to do this was virtually 100%. Nobody was cured by this drug. They had shrinkage, disappearance of tumor, and then it grew right back. So this is, of course, selection. We're selecting for these, the subpopulation that have these mutations. And so these single base changes, of course, are the most common. But the cancer cell population, being genomically unstable to begin with, has all kinds of mechanisms for generating genomic diversity. One of the most common is simply ploidy. You quadruple. Many cancers in their early stages are tetraploid. So now you've got many more genes to mutate to try to adapt to whatever selective pressure is thrown at you in the clinic. So this sort of resistance is quite expected. Resistance to targeted therapy happens because cancers continually evolve. Cancers are a diverse population of cells. Just like any biological population, they evolve and adapt, and that adaptation is facilitated, of course, by genomic diversity, which in the clinic everyone thinks of as genetic instability of cancer. That's why that example I showed you has been replayed with many, many other similar drugs. So our selection pressure, what we do every day in the clinic, is driving us, the evolution of the cancer cell population. It can produce a selective sweep, but if we don't kill every last cell, what we've really done is selected a truly resistant, hard-to-kill subpopulation that eventually grows back up. Now, genetic diversity is often referred to in the clinic as tumor heterogeneity. Now, I think that's a misnomer, but this is the way most clinicians refer to it, not really understanding that what they're seeing is simply a genomically diverse population. So tumor heterogeneity was the unfortunate finding as we started doing genomic sequencing that all cancer cells in the patient are not identical. They are clonal, they have a family tree and a phylogeny, but the extent of branching of that tree is extensive. So from a clinical perspective, it seemed like total chaos, like this Jackson Pollock painting here. No way that you could devise a strategy that could get through this problem if you thought of it as chaotic. And this was really driven home by this landmark paper in the New England Journal of Medicine 5 years ago now, Intratumor Heterogeneity and Branched Evolution revealed by multi-region sequencing. I'll tell you, this is one of the first times I've ever seen the word evolution in the title of a medical paper in the New England Journal. Fortunately, there are now many more of them, but this was a revelation and a shock to many. This was a paper based upon analyzing the diversity of the cancer cells within a single patient. And it was really a technical tour de force. Unfortunately, they could only do this because the patient died of a kidney cancer, and they were able to obtain samples from every deposit of cancer that was in the patient's body. The original cancer that began in the kidney, and then all the metastatic sites, the secondary sites that had spread. And they did genomic sequencing on every one of those sites. Now, of course, if every cell was identical, this map here of all the different clones would look solid. But there were all kinds of variations. These are metastatic sites. These are different sites within that same primary tumor. So what was found were multiple mutations in multiple anatomical sites, in multiple genes. And they were able to draw a phylogenetic tree of how that cancer cell population evolved. You see all the different branches that were genomically different, although all related with this one so-called trunk mutation, VHL. Now, when this happened, this really frightened biotech and pharma that was pouring a lot of money into targeted therapy. That one gene, one drug approach was clearly not going to work when in even one patient you've got so many different mutations that you can't truly characterize that cancer very well. And all of the Wall Street mavens were very worried about this. Stocks took a dive for all the biotech and pharma stocks. But actually, it was not chaos at all. It was a very understandable evolutionary process that had been understood. Because if you look at the phylogenetic tree here of this patient's tumor, they're all related with 100% of them have this gene, VHL, which unfortunately is so hard to target, there is no drug that hits this. And then the branches, of course, had mutations. They happen to be in the same gene, just different sites. Every different branch of the cancer had mutated the same gene in a different way. And of course, we know from evolutionary biology that's convergent evolution. Somehow, that gene is a barrier to survival. Any way you mutate it gets you past that barrier, and every different subclone managed to contain a mutated population that survived the therapy. So it wasn't random. It actually had a logic to it. So if you think about convergent evolution as a basic principle of evolutionary biology, I think it leads you to think about cancer as an invasive species, a diverse population of eukaryotic cells, asexually reproducing, but derived from a complex multicellular organism, and as such has access in its genome to the full toolbox that the eukaryotic cell has. And of course, as you know, all multicellular organisms, multi-organ complex organisms require all kinds of programs during embryonic development that could actually be co-opted by a cancer quite easily. When you think about the migration of cells around the body during embryonic development and the rapid proliferation and growth, all that a cancer has to do is somatically mutate or even simply turn on one of these programs of genes that was present in the embryonic stage. Plus, they're genomically unstable with a very high mutation rate, meaning extensive adaptive capacity. So how do you ever get at a foe like that? Well, I think a lot can be learned from thinking about invasive species in general and the biology of invasive species and all the attempts that have been made with all these invasive species to get rid of them. None of them actually yet too successful. But the principle is, of course, that you've allowed a highly adaptable, well-equipped organism to get into a new ecosystem and rampage through it. That's what most invasive species are. That's what cancer is. I think one of the obstacles to my colleagues embracing this notion of evolutionary biology is the lexicon we use in medicine, because it locks us into certain concepts and ways of thinking, and it implies operating characteristics of the system. We talk about initiation and promotion. We talk about oncogenes and tumor suppressors. They're descriptive, but they don't really explain what is actually, how they're actually driving this population of cancer cells to adapt and thrive. Response and resistance. Well, resistance is really adaptation to the selective pressure. Regression and progression, same thing, the ups and downs of a population. Remission and relapse. These words direct our clinical thinking, and I think distract us from the real biology. So, and I know some of you have heard me speak before, I really think we need a new lexicon. Cancer then is not a cancer, it's a population of genetically related cells. It's a new species invading the ecosystem of the body. And when we talk about tumor heterogeneity, it's simply the genetic diversity within the population, and one can actually measure that. How much diversity is in this cell population versus that one. We actually have evidence that the more genomically diverse a cancer is, the harder it is to treat, the worse those patients do. Instability, of course, is intrinsic to this whole process, but it's the mutational rate that's generating that diversity and adaptability. So in talking about the ecosystem, that's really the tumor microenvironment in which this population is attempting to survive. And our therapies are simply selective pressure on a population. Now, immunotherapy, I like to think of as introducing a predator into this ecosystem. I'm going to talk a bit more about immunotherapy if time allows, because there's tremendous hope. In fact, many people believe, many clinicians believe, that we don't even need to worry about all this genomic diversity and all these targets. Let's just let the immune system do what it does best, which is adapt to all the diversity in the pathogen and hope that we can turn the immune system on enough to simply attack the cancer cell population. I'm going to show you why that's probably not going to work. Resistance is adaptation, and cure is extinction. We want to drive this species to extinction. And we know a lot about extinction. How are species driven to extinction? Virtually every species that's gone extinct throughout life history has had the similar ingredients that led to that problem. Reduced genetic diversity, usually a bottleneck, and selective pressures that exceeded the adaptive capacity of that bottleneck population. That's the recipe for extinction. And the idea would be, can we get a cancer cell population down to a low enough level, a small enough population, where there's simply insufficient mutational diversity to adapt to the targeted therapies that we throw at it? Is that possible? Again, one can go back to the basic principles of how you drive species extinct or how they've gone extinct, because I think it clearly does apply to what we try to do in the clinic. So the implications of all this are that we need to think of the tissue microenvironment where the cancer population is growing as an ecosystem. We need to follow the microevolution of that population over time. And we need to apply what we know about selection and adaptation to try to drive this species to extinction. If cure is our goal, there are people who are advocating a different approach to cancer. Either way, it's going to suggest new strategies for therapy. So in terms of the ecosystem, one can look at ecosystems in nature. So in the riparian ecosystem, so here's a river, and these are all of the organisms that are living near the bank. And they, of course, here have had different adaptations than the organisms living much further away from the bank, where the water is much more available. And you can apply the same principle to a tumor and its circulatory system. So down here, you've got a tumor, and you've got some cells that are very near the blood vessel, that's here. And then you've got others that are quite distant from the blood vessel. And if you're thinking evolutionarily, you would assume that there's actually some difference between them, and not be surprised if your therapy removes some but not all. And here's a graphic of what might be happening. Here's a blood vessel, here's the cancer cell population. The further out they get, the more anoxic they are, the better they're able to adapt to that distance from oxygen and nutrients, while these are very dependent on having high levels of oxygen and nutrients. So if you apply chemotherapy, you apply anti-blood vessel therapy, anti-angiogenic therapy, you will get a big shrinkage in this population, but you'll actually select for this one. You'll come out with something worse than you started with. What about the immune system? Well, the immune microenvironment is all the rage these days, because most of the new drugs are intended to rev up the innate or acquired immunity to start recognizing cancer cells. There is this belief that one of the functions of the immune system is surveillance, to prevent malignant cells from surviving. That would require, of course, that they recognize that the cancer cell is somehow foreign and destroy it. And that does occur with a number of cancers, as judged by what happens in people with immunodeficiencies. If you look at people with HIV, AIDS, or other kinds of immunodeficiencies, or people who have had organ transplants and have their cell-mediated immunity suppressed to avoid rejecting the organ, they do not get the entire spectrum of cancers. If the immune system surveillance of cancer was really that critical, you would expect as soon as you suppress the immune system, you would see the whole spectrum. Lung, breast, colon, prostate. Actually, none of those develop in immunosuppressed patients. What tends to develop are virally driven tumors, like liver cancer related to hepatitis B infection, or certain skin cancers related to HPV, cervical cancer related to HPV. So it is doing some surveillance, but not as much as I think people expect. So in trying to rev up the immune system, if you think of it as a predator that's actually going after the cancer cell population, I think it's useful to think about how organisms try to escape predation. Well, one is camouflage, and that's actually the basis of most immunotherapies. Most malignancies actually overexpress molecules that suppress the immune system around them, making the immune cells think that they're not foreign, that they're just like normal tissue. By uncovering that, there have been some really terrific responses. Another would be simply to outgrow the predator, and that's another strategy that some organisms use. Just produce so many offspring that the predator can't get them all, and that's the source of survival. Another, which I haven't mentioned here, is that since you need some sort of identifying character, some sort of identifying antigen somewhere in the cancer cell for the immune system to recognize it as foreign, if you lose that because it's not necessary, then you escape predation. Or if your population creates so many different antigens that no one comes to the surface as the predominant one, it won't trigger an immune response. So the resistance mechanisms to immunotherapy, I think, can actually be predicted by thinking of it in this way. What about the population genetics of cancer? Well, this is a cartoon of how one does that. This is, let's say, a patient with breast cancer and multiple sites to which it has spread. And when you do genomic analysis of these different sites, all having started in this one tumor in the breast, you can actually draw phylogenetic trees, some of which have long trunks and few branches, or some of which branch very, very quickly. In this realm, people start talking about palm tree cancers versus oak tree cancers in terms of how much diversity there is. So how do you target that kind of diversity, the trunks and the branches? I think it's going to require multiple pathways of attack. So we have evolutionary contingency and constraint, which tells us that subsequent mutations are going to be contingent upon the ones that came before. So it's not chaos, it's not an infinite array of branches coming off of the trunk. The trunk mutation actually decides what the array of possible branches can be, as we saw in this patient. And the branches are likely to have predictable consequences, because if all of the branches that came off of this first trunk required the same mutation, the same gene to be mutated, that must be solving a fundamental survival problem that's helping adaptation. So it's not random or infinite, it's providing a survival advantage, and we are starting to generate an evolutionary rule book. Which branches go with which trunks? In order to do that, you've got to take the cancer cell population and create a phylogenetic tree like this. One of the shortcomings, of course, is that if you only sample a portion of it, you're going to get a misrepresentation of the entire population and apply the wrong drug. So here is the whole forest. This is, in general, what the trees are looking like across different kinds of cancers, and they're not the same. Some have very long trunks, meaning that most of the cancers have the same mutation in their trunk that forms the basis for a therapeutic target. Some of them start branching extremely early, and some have many branches as they go along. This is where cancer research is actually going right now. So here's a few other forests. This is, again, kidney cancer. You can see how many of them have these long trunks with the same gene, VHL. Unfortunately, it's been almost impossible to target that, and that's why there hasn't been a curative therapy yet. Here's lung cancer. Different kind of forest, because nearly all the trunks are different. There's a few recurring genes like EGFR here, but this one has much more diversity in their trunk, telling us that lung cancer is going to be even more difficult to conquer. We're going to have to divide and conquer to really adequately treat lung cancer. Now, this is what happens in lung cancer that has this sort of a phylogenetic tree if you only use a single targeted therapy. You reduce the population that has the target, the tumor shrinks, and then the others regrow. You add another drug on the same pathway, you get some shrinkage, and then another pathway comes up. It's an up and down that doesn't ultimately lead to much extension of survival. So why not target more pathways? Maybe we could hit 3 or 4 of them. I mean, this is the kind of complexity that you see in cancer. You target one step here, and it escapes through another avenue. It's reasonable to ask, can all targets be targeted? How would we design a clinical trial that throws 3 or 4 drugs at one patient? Because if you think about how most cancer drug trials are done, which the FDA tends to require, each drug has to show some effectiveness by itself, and then you combine it with others. It's more of a pyramid approach instead of a concerted, multi-pronged approach. Because some of these drugs will only remove maybe 5% of the population of cancer cells. And the other will remove 10%. So you will get some shrinkage, but not enough to win the approval of the FDA for your drug. People have pointed to the model of HIV therapy. how successful physicians have been in controlling HIV, even without getting rid of the virus out of the body. Simply giving a cocktail of inhibitory enzymes has kept lots of people alive for decades with a disease that used to kill people in months. Well, that's a great model to go after, but I think it's important to keep in mind that although that approach does successfully prevent resistance, the virus only has 9 genes. It's not that difficult to shut down that organism's diversity generation. It doesn't have the eukaryotic genomic complexity of cancer. So how can we use this strategy? Well, there's 2 basic ways. One is to not go for cure at all. One is to coexist with the cancer and simply try not to select the most aggressive biology. Let the population go up and down, and perhaps that will allow the patient to live longer. Diminish selection for the most resistant aggressive biology. Avoid exerting the strongest selection pressure you have, because if you don't kill every last cell, what will come barreling back will be more aggressive and harder to treat than what you had before. That's the thought. So the way to do that is with intermittent dosing, lower doses, to try to maintain a stable population and let the populations compete with each other for resources. Slow attrition rather than massive cell death, because it assumes that we'll never overcome adaptation. So this is called adaptive therapy. A great deal has been written about this. To reduce selection pressure for resistance, instead of using the old mathematical model, let's try to give the maximum amount of drug that stops short of killing the host, but kills as many cells as possible. Makes us feel better to see the tumor shrink away to nothing on a scan, even though we know that there are microscopic cells that are more resistant than before. So this is a minimal cell kill for control. That's the idea. So you maintain a sensitive cell population by adjusting doses. And it comes from so-called evolutionary games theory, where there are trade-offs. The sensitive population competes for resources with the resistant or adaptive population. In many cases, the resistant population has some other survival advantages, which is why it was only a minority subpopulation when the cancer presented. For example, there is a protein that immediately removes any drug that comes into the cell. But it costs a lot of cellular energy to have high levels of that protein produced. So if you give chemotherapy, you induce a population of resistant cells that have that very high level of this P-glycoprotein, but they're very energy dependent. There are other examples in lung cancer as well. So this is what you get when you follow adaptive therapy. You start here with a treatment-sensitive subclone, which is still the majority, and then you treat it, and you diminish the sensitive subclone, and the resistant subclone increases. This is while you're giving therapy. So once you get to this point, you stop, believing that there's going to be clonal interference and the resistant subclone has some sort of disadvantage to the sensitive subclone. You keep treating up and down, but you still end up with a large population of cancer cells here. The approach I showed you with that single drugs in sequence gets you basically to the same place. You shrink one population and another one expands. This is a pruning strategy. You're simply pruning the branches off the tree, but you're not getting the root out. And I like to call this an appeasement strategy. And we all know that appeasement doesn't end well. This is not going to lead to cure of this patient. It may prolong their time on less toxic therapy, but ultimately the cancer will win. So why not be more ambitious? Why not try to drive the species to extinction? To do that, you need to genomically characterize the cancer cell population, characterize the adaptive or resistance pathways, which we are now doing with all that genomic analysis, and then apply multiple therapies that hit the branches, hit the trunk if you've got drugs like that, before adaptation can occur. Create a bottleneck. Even if you can't kill every last cell, if you get down to a subpopulation with a population bottleneck, perhaps that is a less genomically diverse, less adaptable population that you can then go after, perhaps with immunotherapy. Maybe that is the place for the immune system, where the odds are really stacked in favor of the immune cell population against the small population of cancer cells. So I want to show you the example of melanoma skin cancer. That's that very darkly pigmented cancer that develops in some people due to genetic predisposition, in many due to sun exposure. Australia, as you may know, has the highest rate of melanoma in the world because it's mainly populated by exports from the UK, from Northern Europe, whose population was not adapted to the intense sun in Australia. It's a very great example of maladaptation to the environment that this population is now in. So melanoma skin cancer used to be uniformly fatal within months. And then it was found that 60% of them had this one mutation. That's that really long trunk that melanoma cancers have. And when you apply that drug, you get a very dramatic shrinkage. That's what this is here. Big shrinkage. But all patients ultimately do acquire resistance. So let's throw another targeted therapy at it. So when you add a second drug in the same pathway to try to get around how the cancer cell population adapted to the first drug, give them both together, you actually get an even longer period of cancer control. That's with the first drug alone. That's with both of them. So you've definitely made progress. And this is now actually the standard of care along with immunotherapy for this cancer. But it's still inadequate to overcome resistance. So how many targeted therapies do we actually have to throw at this patient to conquer the adaptive capacity of the cancer cell population? Triple, quadruple? Well, here's some insight as to why this is happening and what you need to do. This is that signaling pathway that the drug is hitting. This is that mutated signal that's driving growth. So when you first treat the cancer, you hit that target, and if it's able to escape by mutating around this inhibited target, you add the second one. And that does prolong the period of disease control, but yet they all progress. Why? Well, because you can turn on a parallel pathway by going this way. So if you really want to shut that down, you need those two plus a third, at least, a third drug, to start to shut off all of the escape adaptive pathways of this population. Is this likely to work? Well, this paper actually is one of many where they did mathematical modeling of the likelihood that multiple targeted therapies aimed at these trunks and branches of the cancer population may work. And it was modeled on observations of this very type of patient. So the chance of a single mutation conferring resistance to a single drug is 100%, given the number of cancer cells and the population diversity. The chance of a single mutation conferring resistance to two different drugs is less than 100%. It's still 73%. Still pretty likely that one mutation somewhere in that vertical pathway could get around the two blocks that you just applied with those drugs. The chance that one cell has both mutations is small, though. So what about the chance of a single mutation conferring resistance to three different drugs all hitting different places? It's zero. The likelihood that one cell has all 3 mutations is also 0. So for mathematical modeling, it suggests that there may be a critical threshold of targets that we need to hit that will overcome the adaptive escape. Of course, that's going to be different depending on what kind of tree the cancer cell population observes. So it suggests that some form of combination therapy might be the key and the optimal strategy. You could ask, do it in sequence or all together? And there have actually been some trials already trying this, which showed that if you do it in sequence, as you might expect, you simply allow time for the adaptive clones to expand. So better to have an educated guess as to which targets you need to hit, and try to hit as many as you can all at once. So predict adaptive responses, catalog the recurring mutations, anticipate the resistance pathways, and then perhaps come in with an immune-based therapy to try to reach the remaining population that's too small to even analyze genomically. Now, as far as the cataloging, that's been done and being done. This is the Cancer Genome Atlas that sequenced 12 different cancers on many different levels, not just genomically, but also at the level of RNA expression, methylation, protein copy number, gene copy number, etc. And I like to think of this survey of the world of cancer and basically producing a taxonomy for all of them as our own kind of voyage of the Beagle, where we're really cataloging the genomic anatomy and variety across the span of cancers. And this is the kind of data that's being generated. You see, this is this year. What are the top mutations in lung cancer, pancreas cancer, kidney cancer? Some of them are the same, but a lot of them are different. So the catalog, the evolutionary rule book, is being generated. And even the trunks are now understood. These are standing for different kinds of cancers, and we now know if you're a melanoma, these are the most likely trunk mutations, and if you're a lymphoma, these are the most likely branch mutations. So the catalog is being filled out. It's just a matter of using it. So we have the catalog, we're getting that. We need to effectively block those pathways, and we need to be able to give targeted therapies together in a safe way. Probably rapid sequencing or combinations. But what about the bottleneck population? What about immunotherapy? There's tremendous hype about immunotherapy for good reason. There have been significant reductions in important hard-to-treat cancers with immunotherapy, turning on the immune system to attack the cancer. And people like the idea. A lot of clinicians who aren't particularly enthusiastic about genomics or comfortable with it really like the idea of immunotherapy. Let's just let the body fight cancer. All we need to do is rev up what's already there. Immunotherapy, a revolution against cancer. Despite all that hype, if you actually look at the data, the big responses that are occurring in lung cancer, kidney cancer, melanoma, it's only about 1 out of 5 patients where you're actually able to rally the immune system. The rest don't respond. So is it the answer to cancer? Well, I think it is an answer, but it is not what some of my colleagues think, which is an alternative to targeted therapy. Many people say, let's not even bother doing genomic analysis of cancers. Where is it getting us? The diversity is beyond us. Let's let the immune system adapt the way it's supposed to. There is strong selection on the cancer cell population for immune escape. The tumor antigen that the immune system has to recognize needs to be necessary for survival, or it'll get sloughed off as an adaptive response. A non-essential tumor antigen will be lost or mutated away, and no longer recognizable by the immune system. And there have been many instances now of acquired resistance to immunotherapy and a minority of long-term responders, as I said. So might it be most effective for a bottleneck? Well, this is a fanciful approach where I think you can treat a cancer cell population with multiple targeted therapies and bring it down to a single bottleneck that, if you don't have a way to get at it, will expand once again. This would be the group of cells within the population that you would like to have an immunological response. So, to conclude, and thank you for your attention, I think it's time for the clinicians and the cancer researchers to really think of cancer as an evolving population that's following the same principles as any population of organisms, following the principles of evolutionary biology, whether it's a herd like this or a herd like this. It's going to be the same biological principles. And I think you've heard me say this before, any of you who heard me speak, but as far as the ultimate goal of driving cancer to extinction, I think it's safe to say that no force of nature apart from an extraterrestrial asteroid has been more efficient at driving species to extinction than we have. We're very good at driving species to extinction, so we certainly should be able to apply that skill to the problem of cancer. Thanks for your attention. Yes?
Don't you think that we'll probably never get rid of cancer in some sense? I mean, we can deal with individual populations of cells, we can drive them to extinction, but there'll always be more every generation, every person.
You mean that cancer is always going to develop in individuals, even if you get rid of one cell population, it might arise in a different place?
Yeah, I mean, I guess what I'm just saying is that I've seen some commercials where they're talking about how they're going to end cancer and destroy cancer, and they're going after, first of all, cancer doesn't care, it doesn't have opinions, emotions, but besides for that, I don't think we're going to get it out of our genome. I mean, doesn't every species have cancer, or every complex, I don't know which level in biology, but certainly mammals, don't they all have cancers?
I certainly agree with you. I don't think the problem of the development of cancer, carcinogenesis, is ever going to go away because it's an inherent risk of a multicellular organism with imperfect gene copying when cells divide. That's the price for being able to evolve and adapt, having some genomic diversity and the risk of mutation. And if you hit the lottery and you've mutated a critical cancer-driving gene, that cell population is on the way. I think stochastically, it is happening all the time, but there are very highly evolved DNA repair mechanisms to try to shut it down. And we all know, of course, the things that we do in our own lives that might tip the odds one way or another. So if you inhale a lot of carcinogens, you're going to increase the likelihood that one of those populations is going to make it to be a clinically evident cancer. If you believe that you're reducing your intrinsic oxidative stress to the DNA by eating a lot of antioxidants and avoiding oxidizing exposures, maybe you can lower the odds a bit. But I totally agree with you. I don't think the problem of cancer will ever disappear. When they say we're going to destroy or eradicate cancer, I think what they're meaning is we're not going to let it take lives. We'd like to not let it take lives the way it does now, if it can be more effectively treated than it's been. Because even with all these advances, the majority of people with cancer, if they're not cured by the surgery, are going to end up requiring toxic drugs like chemotherapy or hormonal therapies. So I still think the future is in these targeted therapies, but we're not going to eradicate the basic problem of that carcinogenesis process. Yes?
So I want to pick your invasive species analogy apart and think about the bottleneck problem. And it strikes me, just looking at the ecosystem of the Santa Monica Mountains here, the events that we've seen over the last 6 years or so, where we had a prolonged drought, there are a number of invasive plant species that are reliant on a steady supply of water in a way that the indigenous species are not. And they escape from people's yards where they have sprinkler systems, and they get into stream beds and riparian areas, and they do well there, right? And they displace the indigenous species. But then, in the presence of a prolonged drought, they're eliminated, and the indigenous species come back. So, the analogy there is that if there is, if you can introduce a resource upon which the invasive species is more dependent than the cells of the host in this case, right? And then withdraw it, potentially you can use selection to your advantage. So if you create the bottleneck that you've described, right, and then and my cell biology is elementary school level of this, so I don't know what that required resource is, right? But if you provide it in abundance such that you're favoring selection within the residual population, where the variants that thrive but are dependent on that resource, akin to the exotic species that are dependent on the sprinkler system in the backyard, right? And then withdraw it, you use selection against the tumor by favoring the growth of the dependent species and then removing the resource. Mm-hmm. Is that just fantasy, or are there possibilities?
Well, I think it certainly makes sense, and there have been instances where a growth-promoting substance has been given to try to let a subset of the cancer cell population become dependent on it and then withdraw it. The best example of that was with breast cancer, actually, where for some time, this was a few decades ago, one of the therapies was to actually give extra estrogen, because that most breast cancers were using estrogen hormone even in tiny amounts to grow, and then pull it away. And that actually would cause a great deal of cell death. But it's still a rather severe selection pressure. So unless you can be sure that there isn't a remaining cell that has managed to survive without it, you're still susceptible to having that recurrence. But I think it's a very valid approach. I think you can start to do that if you characterize what it is they need. If you know, for example, that VHL mutation that's so common, so ubiquitous in kidney cancers. Well, that's intimately involved in the tolerance of oxygen deprivation. And so the tumors can grow big with having little oxygen penetrate because they've got that mutation. But they can only go so far, and then they have to mutate another gene, that other one, SETD2, which is actually related to genomic diversity. It actually deals with the turning on of a whole variety of genes, like opening a whole new toolbox to adapt to the expanding tumor. So I think that approach is going to be informed by the specifics of what's driving that cancer. Yes?
Thanks for a great talk. I had a question about the metaphor of invasive species. It seems like all of these approaches, which sound really promising, are all trying to eradicate the invasive species once it's already made it to the Great Lakes or whatever. So I'm wondering, it seems like there's a detection side to this too, right?
There's a
detection. Like, detecting, I mean, are there insights about detection that would help to?
Yeah, so detection is another, not only detection of an unsuspected cancer that is not seen or detectable by any scan or physical exam, but by a blood test, for example. There's the ability to detect subclinical cancers, and then there's the much broader area, which is to detect the remaining subpopulation that's simply too small to see. And they're doing that with so-called liquid biopsies, where if you know that within the primary cancer there was a mutation in a critical gene like EGFR, growth factor in lung cancer, and you know what you're looking for because you found it in the original cancer and then it disappeared, people are now doing so-called liquid biopsies, which is simply drawing blood. Because as it turns out, some cancers will actually spill DNA directly into the bloodstream that can be detectable with current genomic technology. The problem with that is that you have to be looking for something specific. You can't just go fishing. You've got to know which particular mutation you're looking for. But that's how some are approaching this. I showed you that curve of the single drug therapy against lung cancer where the population goes up, you treat it, it goes down, it comes back up again. So instead of waiting for it to come back up as judged by appearance of a tumor on a scan, they're starting the next therapy at the first sign of the mutation in the bloodstream, before it's big enough to see. So the population will be a lot smaller by definition. And I think if you do that with more than one agent, you may actually get really prolonged periods without regrowth. Perhaps you will hit enough targets that you've killed every last cell. But that's a huge area now of biotech business, is offering liquid biopsies. Because nobody wants, you can't go in and take pieces of tissue to monitor what the cancer cell population is doing over time or how it's changing. No one will subject a patient to that many biopsies. But if you can detect in a tube of blood whether or not you've eradicated that clone, that's very promising. And so there are many companies out there, many labs that are offering that. Yes?
One thing that occurred to me when you described cancer as a population, I don't know if this is something that already exists, but would be rather than targeting the cancer itself, what if you were to target all the healthy cells and try to provide some sort of advantage of extracting resources that are available or something of that nature, or even inducing some kind of antitoxin kind of thing to the healthy cells and then applying a more general toxic element? Is that something that sounds could exist? Or
yeah,
does
so
exist?
you're describing the strategy of targeting the so-called microenvironment rather than just the tumor cell itself, attack the normal cells, or at least make the environment less hospitable for the cancer cells. That's the basis for the so-called anti-angiogenic therapy or anti-blood vessel therapy with drugs like Avastin, for example. There are quite a few drugs out there that work not against by targeting anything in the cancer. They're targeting the cancer's ability to induce blood vessels to bring it nutrients. And the problem that happened is the one I actually illustrated to you, which is that you're selecting for the subpopulation that's able to adapt to the diminished access to nutrients. And what was seen was that as you give these anti-angiogenic therapies, you get some initial shrinkage, and then you get rapid growth of a different subclone. And it probably works in combination with other things, but so far it hasn't. What else could we target? Well, there are chemicals that attempt to prevent oxygen from being utilized by by cancer cells. But that, of course, is very hard to separate from normal tissue. Any other questions? Yes?
Has there been any attempt to sort of design a multi-drug trial in this way? Because you have to sort of effectively change the entire process of a drug trial.
Yeah. Well, it gets back to whether or not one has to prove that each drug individually has its own activity. It appears that the FDA is starting to relax that requirement a little bit, and there are 3 drug trials, although they're rarely 3 targeted drug trials. They're usually chemotherapy plus 2 other things. Now there are immunotherapy trials plus 1 or 2 other targeted therapies. That's actually the design of many trials now. But most of them had to show their own intrinsic responsiveness. And I think that's probably a mistake, because there are certain inhibitors that have a synergy. Neither works very well by itself, but if you give them together, you really get such synergy that you can give less of each and achieve the same biologic output. The trials that you're talking about are mainly being done in animals. They're being done in mouse models. But how well that's going to extrapolate to the human condition, I think a lot of people don't trust it. Yes?
I have a sociological question, evolutionary biology one, which is, you mentioned earlier in our private discussion that you're traveling the country a lot, speaking to oncologists in another capacity that you hold. But I'm curious whether outside of that capacity you have conversations of this nature with them, and if so, what the reception it receives.
Well, I try to use the words. Instead of saying resistance, I try to say adaptation. And instead of saying the cancer, I try to say the cancer cell population. So I'm trying to use a different set of words, the lexicon, to have people conceptualize things differently. Because what I am out talking about, of course, is a gene expression test that can predict the behavior of breast cancer, and then determine how much therapy to give or how aggressive the therapy needs to be. But then I'm always asked, well, what about this patient? We treated them this way. So much of it is the ups and downs of the population. It's just being chased. You're always a step behind it. So I think most oncologists think of evolutionary biology as an interesting sort of intellectual exercise, and some of them do accept the mismatch maladaptation notions. But as far as directing therapy, I haven't really seen it seeping in to the way people think.
Just following up on that relatedly, so I mean, there's been a lot of work on evolution of antibiotic resistance, and presumably clinicians are intimately familiar with that problem, albeit perhaps across specialty lines. I mean, does that provide a kind of proof of concept of some of the core conceptual issues to people in the clinical world?
Yes, I think the majority of uses of antibiotics, though, are single drug therapy, because the goal always is let's culture what we're treating so we know what we're treating, and then the in vitro determination of sensitivity is very accurate with bacterial pathogens. So if it tells you it's penicillin resistant, but it's sensitive to a different drug, then you're directed that way. And that was tried with cancer, to grow it in the test tube and throw drugs at it and see what killed it. And it simply didn't work, partly because what you get from the tumor and try to grow may not be representative of the whole population. That's that thinking. Doesn't matter where you sample it, they're all the same cell. Obviously they're not. But it's rare that we use drug combinations. Yes?
In the slide where you showed the differences in population morphology and variants, do you know if there's any dramatic differences based on whether or not the cancer originated in that piece of tissue or as a result of the cancer spreading to a different stage and it's a result of another type of cancer?
Well, I think if you're asking, is there variability between the cancer in its primary where it started versus landing in some other part of the body? Yes, absolutely. Some of the differences are not mutational, but gene expression differences. There are certain genes that seem to get turned on or off to help a metastasis survive in the liver versus the lung. There may be even some homing mechanisms which explain why some cancers preferentially metastasize to bone and never go anywhere else. There's something about the microenvironments there that are more hospitable. There may be growth factors that particular cancer cell population can use. So the expression array is actually quite different if you look at the secondary deposit in a different organ versus the primary. But the mutational pattern isn't really that different. You can trace the lineage quite well with those phylogenetic trees. Yeah?
So the immune-based therapies that you said have a little bit of success, but not overwhelming success. Do those work by kind of cranking up the full immune system, like turn up the power on the whole thing, or are they somehow targeted? Like, can you train the immune system to selectively respond to certain cancers analogously to vaccines training to respond to certain pathogens?
So the big news last month was with something called CAR T cells, C-A-R-T, and this is a highly engineered kind of T cell in the immune system that is induced to respond to a particular antigen that was found in the cancer of interest. And there were news stories about I think children with brain tumors that had great responses, or people with lymphoma, because that's the easiest to treat with that approach. That's incredibly technology and labor intensive to do. And if you have one tumor antigen, it'll work. But as I said, if the tumor antigen isn't essential for the survival of that cancer, it's going to be lost as an adaptive strategy. The other immune therapies, which aren't cellular based, I mean, these are cells that are actually infusing back into the patient after they engineer them, at a cost, by the way, of nearly $500,000 per patient. Doesn't sound like a healthcare solution for the United States. The other is to just give an antibody that homes in on that camouflage receptor, so-called PD-1, the thing that cancers express that makes the anti-cellular immune system not recognize them as foreign. So if you cover that up with the antibody, the immune system suddenly recognizes the cancer and kills it. And those are the responses that are 20, 30%. The downside is that when you do that, you also expose some normal tissues to attack as well. So the main toxicity is autoimmunity. The immune system is unleashed to attack the lungs, to attack the GI system, to attack the heart. And that's been one of the limiting factors to somewhat non-specifically trying to rev up the immune system.
Okay.
Great questions. Thank you. Thanks for your attention.